Now, tumor can be of two different types. While one type of tumor where the cell is dividing and producing undifferentiated mass of cell, but that tumor, those cells lack the quality of spreading itself from one location of the body to the other location of the body. Okay, but the other one, which can transfer from one place to the other place of the tissue, is known as malignant tumor, and that is a part of cancer biology. Okay, so cancer is nothing but uncontrolled growth of cell which ignores all the signs and symptoms of cell growth stop or cell apoptosis. Okay, so let's talk about cancer biology in this chapter, in this class. Okay, so to understand cancer biology very well, you need to know that cell cycle regulation stuff clearly. Okay, that's why we talked about it. So first of all, the formation of cancer is known as oncogenesis. It's a molecular term, oncogenesis, or we can also call it transformation, because the word transformation in prokaryotes mean transferring DNA fragments from outside inside the bacterial cell, while transformation in eukaryotes mean this conversion of a normal cell into a cancerous cell, also known as oncogenesis. Genesis means the generation or start point of oncogene, this conversion of normal into oncogene. Okay, so oncogenesis is more than one event. It's not a single event, it's multiple event together. Okay, means it needs to have multiple gene mutations to cause cancer, right? So generally what I should say, in any type of cell signaling pathway that we know, we can divide them into two different parts. A pathway that will allow the cell to die, a pathway that will allow the cell to live and divide. So if the pathway allows the cell to divide, and if that pathway is upregulated uncontrollably, and the pathways that cause the cell to die is not working properly, then what will happen? Then the cell will divide more and more, ignoring the sign of apoptosis. Then the cell may turn cancerous, right? So for cancer, it's a multi-stage process. We'll be seeing the different steps, and also for cancer you need more than 2-3 types of mutation to convert a normal cell into a cancerous cell. Only a single mutation is not enough. For example, here it's a normal cell. Let's see, the first mutation is there. After the first mutation, let's say the cell seems normal but is slightly altered its behavior in terms of their growth. Then a second mutation, the cells start to proliferate, but other things are normal. Then the third mutation, cell proliferates more rapidly, and also cell is undergoing structural change. And fourth mutation, and any other mutation after the fourth mutation, ultimately turning that cell into malignant cell, because to become a malignant cell, this cell need to alter multiple behavior. One of such important behavior is metastasis, and second property is angiogenesis. These are very much unique feature of cancer cells. Okay, metastasis and angiogenesis. Metastasis is the property with which the cancer cells can move from one place to the other, one tissue to the other tissue of our body. And angiogenesis is the one where cancer cells start forming their own blood supply, because our body has natural way to prevent the conversion of normal cell into a cancer cell. Because while a cell is dividing, one cell divides into 2, 2 into 3, 2 into 4, 4 into 8, continues to divide. There is a situation, there is a property of cell known as contact inhibition. Means while neighboring cells, while start dividing, they are contacting each other. That will prevent the further growth of that cell. So there is a limit, an extent till which the cell will divide. Once they receive, let's say there is a wound, and due to the wound some cells are dead in the middle of this tissue, rest of the cells are there. So now new cells start forming there, and those cells start dividing until they interact with the neighboring cell, because while they interact with the neighbor cell, due to this contact they know that yes, there are cells present, so they don't need to divide anymore. So this is known as contact inhibition. Now cancer cells never follow this contact inhibition, because although they have cells nearby, the cells continues to divide. Now the multi-stage process begins with stage 0, 1, 2, 3 and then finally 4. Stage 0, where just there is a creation of cancer. Now what we mean by this particular stage? Creation of cancer means there is a mutation. The primary mutation that drives one type of cell to become cancerous. Okay. Now stage 1, where the cancer cell, although there is a small modification of the gene, mutation of the gene, but it's restricted to one part of our body. So cell is not transferred from one place to the other. Now stage 3 is a situation, stage 2 is a situation where the cancer cells start to achieve metastasis feature. So they start to migrate to the local regions of their tissue. Then stage 3 is locally advanced, so the cells start migrating little far, and then stage 4 is the final stage where the metastasis is totally done and the cell can now spread from one location to the completely different location. That means from respiratory system to our intestinal system. So they can move from one place to the other, far long distances across the body they can reach. Okay. So as I said here about the contact inhibition, this is a normal process to prevent the unwanted cell growth and cell division. Now cancer cells ignore this sign. Second sign that cancer cells ignore is that another problem a cell will face who is rapidly dividing, trying to divide fast and fast, that lack of, they will get the lack of supply of nutrients and supply of oxygen, because if they have lack of oxygen or nutrient, the cell will not divide, because they are dividing more and many cells are accumulating. But the problem here is, in case of cancer cell, they also face the same situation, but they can handle it. How will they handle? Is that they can influence our blood vessels to grow nearby, so that they can continue to get the supply of oxygen and nutrients. Okay. So as I said, the different stages of transformation. Let's say the first mutation is in the Apc gene. Now what is Apc gene? Adenomyces polyposis coli. It's a gene, mutation of this gene this is a tumor suppressor gene. So mutation of this gene, and if this gene is inactivated, then the cell may turn cancerous. Let's say this is the first mutation to a normal cell. Now although a tumor suppressor gene's job is to prevent cancer, but now the gene is not functioning anymore. But this one mutation is not enough. Now it gets the second thing, second mutation of KRAS mutation. RAS, these are oncogenic proteins. Okay, so once you mutate KRAS, then what happens? Then the small modification, now second mutation and upregulation of the KRAS. So KRAS is producing more and more, that will cause and allow the cell to divide and grow and more. So as a result, there will be rapid growth of the cell and formation of tumor nearby. So at this point of the time, we call the tumor cells as intermediate adenoma, because earlier, before just one mutation, we just call it as a normal cell, and after one mutation, hyperproliferative state or early adenoma, and then after the second mutation, we call it intermediate adenoma. Now it comes a third mutation of a gene known as DCC gene, deleted in colorectal carcinoma. This is the name of the gene, and this is a dependence factor. So again, mutation of DCC gene causing the intermediate adenoma to turn into a late adenoma. And late adenoma is very much alarming, because now that is the time point where the cell is acquiring all the process of metastasis and stuff. Now finally, there is another final mutation, in this case P53 mutation, will turn the late adenoma into carcinoma. We can only call stage of cancer as carcinoma when the cell receives all the hallmarks of cancer. Hallmarks of cancer means all the properties of cancer that we are talking about. For example, metastasis, angiogenesis, all the properties now are fully fledged and they are formed. So now we can call it, now this is carcinoma. So once carcinoma is caused, then what will happen? Now other mutations, and it will turn and metastasis, and moving from one tissue to the other and doing the same job in the new tissue while they reach there. Okay, so you can see here point mutations are very, very dangerous. So point mutations are one of the leading causes of cancer, and the point mutation means mutation in one particular nucleotide, and that may happen all the time due to the different gene modification of important genes. And there are basically two types of genes in terms of cancer. One is all these tumor suppressor genes, and the second type are the oncogenes. Okay, now apart from point mutation, there are also chromosomal changes, chromosomal structural changes that may also lead to cancer. An example for such here we can say translocation, deletion and insertion. These are the different modifications that may lead to cancer. For example, if we look at here translocation, let's look at the first type, translocation. So what happens is that the chromosomal translocation of chromosome 8 and 14, swapping of the chromosome segment between chromosome 8 and 14 may lead to formation of Burkitt's lymphoma. Okay, it's a cancerous situation. Now why is translocation can cause cancer? Try to understand the mechanism. Now chromosome number 14, there is a gene immunoglobulin H or IgH, and immunoglobulins, they are housekeeping genes, and we need to produce them continuously all the time, no matter what we need them or don't, but we need to produce them always. In a very basal level, the expression is always on. And chromosome number 8, there is a gene MYC, c-MYC, all these MYC genes are oncogenes. So normally the product of MYC somehow help the cell to grow and divide through the cell cycle. Now what happens here is the fragment of chromosome 8 containing MYC is transferred near the promoter of immunoglobulin H in chromosome number 14. And we know promoter for IgH is it's a constitutive promoter. So as a constitutive promoter, it will allow the MYC to be translated always, okay, in a constitutive way, in a synthesizing MYC. So as they are synthesizing MYC in a constitutive way, then what will happen? The swap between 8 and 14 may lead to the formation of tumor, because the cell will continue to divide and they will divide fast and fast. That will lead to the cancer, okay, or Burkitt's lymphoma. That's translocation. Second situation, let's look at deletion. Now deletion may also lead to behavior of normal cell into cancer. How exactly? Let's look at here about a protein PRB. RB known as retinoblastoma sensitivity protein, and we all know about the role of PRB. What is the role of PRB? PRB actually blocks E2F. So in turn, PRB is kind of tumor suppressor gene. It's preventing E2F, which is a transcription factor for cell cycle to progress from one phase to the other. We saw that in case of the G1S transition, right, if you recall. Now what happens here, as this is a normal chromosome number 13, and the normal chromosome number 13 with both the locus PRB is active, making the protein, so no problem here, normal cell. Now in a situation of retinal cell where the chromosome number 13 is deleted, some fragment of chromosome number 13 is deleted, and that fragment carries retinoblastoma sensitivity protein. So as that fragment carries retinoblastoma sensitivity protein and it's cleaved away, then what will happen? That chromosome number 13 is no longer producing retinoblastoma sensitivity protein, as without the PRB the E2F is always free. E2F is now unregulated, it's always free. As E2F is free, it will cause the cell to grow and divide always, okay, causing cancer. Now third situation, third is insertion. Insertion, this causes the MYC to be constitutively expressed. Okay, how? So here you can see a c-MYC proto-oncogene with multiple exons. Okay, in the position, we are not actually looking at RNA, we are looking at DNA, but in terms of, where are the position of the exons and introns. So these are exons that are pressed. So this is c-Myc proto-oncogene, and they may have integration of other DNA element from the virus, like avian leukosis virus here, ALV, avian leukosis virus. The integration of avian leukosis virus right in between exon 1 and exon 2 will cause the shifting of the exon, because you can see that exons now are separated due to the addition of ALV. Now as a result, what happens that the read frame now becomes from only exon 2 and 3, not from exon 1. And in this case of the c-Myc proto-oncogene, there are 3 separate exons coding different parts of the protein. Exon 1 is providing the inhibitory element or regulatory element of the protein, while exon 2 and 3 together become a part of the activator unit of the protein. So now as exon 1 is not being translated due to the interruption of this exon 1 and 2 in between the places, as a result only the activatory element is produced. So there is no regulatory subunit attached with c-Myc. So c-Myc is working in an unregulated fashion, and c-Myc is an oncogene, so we know that ultimately c-Myc will cause the cell to grow, cell to divide. So that is another alarming situation and causing cancer, right? Okay, so now we will talk about 2 important features of the cancer cell. As I told you earlier, metastasis and angiogenesis. Metastasis is spreading of cancer cells throughout the body. Okay, an environment where tumor cells can proliferate, invade in the surrounding tissue, and then released into the circulation, invade a distant organ, and establish their own blood supply is known as the angiogenesis. Now, angiogenesis is one of the most dangerous feature of cancer cells, because that makes cancer even worse, because we can detect cancer, we can detect by checking the molecular hallmarks of the cell to tell whether the cell is cancerous or not, or we can easily tell whether the tissue is behaving cancerous or not. But the difficult part, the problematic part here will be like this metastasis, because even though we can tell which part is cancerous, which part is not, whether the cell is cancerous or not, but still if the cell is getting the feature of metastasis, then we will never know whether the cell is transferred from one place to the other. So although we can detect the cancer, let's say we detect the cancer in the lungs, but we'll never make sure that the cell has not started the metastasis. That's the problem, because that's why when we find out cancer, we not only find out in one particular place, we can find out in multiple places of the body at once. Now, how metastasis is actually achieved? For metastasizing, the cancer cells need to destroy tissue, destroy the tissue cementing material, destroy the tissue cementing lining. So how exactly they can do it? So with the help of UPA, urokinase plasminogen activator, and collagenase enzyme. Collagenase enzyme, they degrade collagen protein. Okay, so what happens here is that urokinase plasminogen activator is secreted by tumor cells. Tumor cells also secrete procollagenase. Now this urokinase plasminogen activator, or UPA, convert plasminogen into plasmine, and then plasmine along with angiotensin together, what they will cause a conversion of procollagenase into collagenase, which is the active form of the enzyme, and collagenase can easily do the proteolysis of the basement membrane and extracellular matrix material and break the extracellular matrix, and then can easily squeeze through that place to the surrounding locations. That's how they establish the fact of metastasis. Okay, so 2 things together, UPA and procollagenase secreted, and the rest of the job is done. Now, how angiogenesis is occurred? Because as I said, without the proper supply of nutrients and oxygen, these cells may die. These cells may not survive. The cancer cells may not survive. But here, cancer cell is easily surviving because it's causing the formation of new blood supply. So for example, here we have these 2 release this cancer cells as they're forming tumors. They start secretion of vascular endothelial growth factor and fibroblast growth factor. Both of these, vascular endothelial growth factor and fibroblast growth factor, especially VEGF, it causes the blood vessel to be formed towards the direction of tumor. Okay, causes the blood vessel to be formed. So blood vessels are being introduced. Okay, and it's a chemotaxis mechanism with which the blood vessels slowly start to move towards the tumor cell, and then the blood vessel is kind of interacting with the tumor cells, cover the tumor cells totally, and then the tumor cells can start receiving the oxygen and oxygen and nutrients from the blood supply. Cancer and angiogenesis. As I said, tumor cells releasing vascular endothelial growth factor and fibroblast growth factor. It does all this job. Okay. Although here it's one thing is mentioned wrong. So ultimately, in a molecular way, if you look at the cancer as a molecular event, there are only two types of genes that are dealing with the growth of the cell and the death of the cell. The cell growth pathways and cell death pathways. This is oncogene and tumor suppressor gene. Oncogenes are those who allow the cell to grow and cell to divide, who inhibits apoptosis, promotes immortality, promote angiogenesis, promote metastasis. And the last three properties are very much important properties of cancer cell. While the second type of cell are tumor suppressor gene. Tumor suppressor gene induce pathways which inhibit the cell division, promote apoptosis, inhibit immortality, inhibit angiogenesis, inhibit metastasis. Just the opposite of what oncogenes can do. Now, in a situation, or in every situation of cancer formation, tumor suppressor genes should stop functioning and oncogenes should start unregulative function. Then only the cancer will form. If either one of the situation is working, cancer will not occur. Because if oncogenes are growing much faster rate and uncontrollably, then tumor suppressor gene can prevent them and prevent their function, because tumor suppressor has overrule and override switch over oncogenes. But that's why in most of the cancer, above 98% of the cancers, a tumor suppressor gene mutation is linked with it. And above 90% of all the cancers dealing with, we have P53 mutation as a common factor in all of it. And P53 is probably the biggest tumor suppressor gene available. So this is a list of proto-oncogenes and products that they regulate cell division and apoptosis. So you should remember all of it, because they can ask you questions from their short questions and broad questions. They ask a lot of questions from P53 MDM2 correlation, as well as the list of oncogenes, as well as tumor suppressor genes and how they are interacting. By not telling you the name of the exact protein, but they can tell you different situations and experiments or hypothetical conditions, and after reading the question of group C, you need to tell which is correct. So for example, this tumor suppressor genes can be of different type. In this case, these are all oncogenes. So oncogenes can be of different type, like growth factor. It can be phosphatase enzyme. It can be transcription factor. It can be growth factor receptor, like PDGF, FGF. These are oncogenes. PDGFR, EGFR, oncogenes. Adapter proteins like GRB2 involved in RAS MAP kinase pathway, oncogene. Phosphatases like CDC25, it's activator, that's why activator of cell division and cycle, that's why it's an oncogene. Transcription factor like MYC, JUN, FOS, E2F, all oncogenes. So these are oncogenes, they activate the cell division, and there are also oncogenes who are activators of apoptotic resistance. And remember this term, apoptotic resistance. Apoptotic resistance means in turns survival, right? Example, BCL2, MDM2, NF-kappa B. These are all part of apoptotic resistance proteins. Okay. Now, a final question regarding this transformation. We've seen the transformation in terms of mutation. Point mutation can always cause cancer. We have also seen that the molecular changes like the chromosomal changes, structural changes like translocation, deletion, insertion can also cause cancer. Now another causing agent of cancer is virus. Now it's a big question how exactly virus can cause cancer. Now virus like human papilloma virus is a leading cause of cancer, especially cervical cancer. Approximately 50% of the women infected with this human papilloma virus based cancer, and they form like benign skin warts and genital warts. In United States the documentation is given, it's approximately 10,000 women and 4,000 deaths every year. Now the annual case report is also shown, and mostly they cause is mouth, okay, and cervical cancer, and the HPV induced cervical cancer is almost all of it. Now how exactly HPV induces uncontrolled proliferation of the cell? Now obviously they need to change either molecular terms or the behavior of the cell. How exactly? The HPV virus once transfer its genetic component. That genetic component contains different fragments, E2, E3, E4, E5, E6, E7. Among them, this E6 and E7 are the two major components, two major genetic elements that involves in causing and turning the normal cell into a cancerous cell. How? Because E6 inactivates P53. If you look at here, E6 inactivates P53. So as E6 inactivates P53, then what will happen? If P53 is inactivated, then the cell will not grow, the cell will not divide. The cell will continue to divide. I said totally wrong. The cell will continue to divide. Even situations where there are damage and stress situations, the cell continues to divide because there is no guardian to stop them, right? And then E7 inactivates PRB2. Remember, PRB and P53 both are tumor suppressor genes. And these tumor suppressor proteins, if the tumor suppressor proteins are inactivated, then what will happen? Then the cell will divide and grow uncontrollably without any kind of pause, and that is a dead alarm. Okay, now let's talk about the cancer treatments. And remember one thing, cancer and any other pathway related to cancer is very much important. So as the treatment part of the cancer, they ask question from cancer treatments now and then often, right? Okay, so what we need to know here is any drug that is associated with cancer treatment and their mode of action, like where exactly the drug is acting, and the name of the drug. That's all that we need to know. So I'll tell you few of the drugs and where exactly we're using it to prevent cancer. First of all, we will talk about the different types of cancer treatment, and then we'll talk in details about the drug treatment. So there are three types of cancer treatments: chemotherapy, radiation therapy, and drug therapy. Chemotherapy involves DNA mutagens. Okay, radiation therapy means ionizing radiations or X-ray. These are the things. And drug like Velcade, Herceptin, these are the drugs that are involved. Okay. So now the question is, in all these cases, what is the difference between chemotherapy and drug therapy? Chemotherapy is injection and drug therapy is not only that. Chemotherapy and drug, the difference is that in drug and chemotherapy both we are using chemicals, but in chemotherapy all those chemical agents are direct mutagens. They are direct mutagens. DNA mutagens. So they will change and mutate the DNA, reversal of the mutation. That's what we are trying to do with chemotherapy. While in drug therapy, the drug target is not the DNA itself. The drug target are different. Got it? Because as we know that mutations cause cancer, we can use the reversal of the mutation by the mutagens. So first of all, let's look at the different types of drug therapy, because that's why we are more interested in, because the chemotherapy agents are always targeting DNA, fix the mutation. Okay, but the drugs, they have different target. So first type of drug here Velcade or Bortezomib. Velcade or Bortezomib, the first drug targeting proteasome. Okay, first drug targeting the proteasome. And you know, proteasome is a very interesting and important cell organ in the organelle, in this case of cancer development. Because remember, while we are discussing about the cell cycle regulation and cancer, how many times we have used the term proteasome and ubiquitin mediated degradation? Almost every single time. That's how important the role of proteasome is during the cancer spread. So without proteasome, cancer cells cannot produce and accumulate all their necessary proteins in their proper amount. Because with proteasome they can break down proteins, they can break the large proteins into fragments, and they can reuse it, which is not possible without the proteasome. So in this case, proteasome is prevented. I mean, proteasome is blocked with the help of the drug. So we know proteasome has two different channels, an entry port and the exit port. Entry port is the place through which the protein, which is tagged with ubiquitin, takes entry into the proteasome, just like a chamber, and there it's destroyed and the fragments of the protein removed from the exit channel. Now the entry channel of the proteasome complex is blocked by the drug Velcade. Okay, so as a result, proteasome is not functioning, so there is no way that the cancer cells can utilize the proteasome for conversion of normal cell into cancer. Drug number 2, p53-MDM2 inhibitor. p53-MDM2 inhibitor, and the example is Nutlin-2. Nutlin-2. Synthesis of E3 inhibitor. What is E3? So let's look at here ultimately. E3 is also linked with the ubiquitin pathway, but Nutlin-2's job is to bind to the p53 pocket, inhibiting the MDM2 binding. Try to understand this pathway. We know that p53 is always produced in our body. Now it is MDM2 that's preventing the p53 to function, and it destroys the p53 always. Now this interaction is very much important, so that the p53 is, the concentration of p53 is regulated in our body. But now as the drug interferes with the binding of MDM2 with p53, p53 is always accumulated in our cell. High accumulation of p53 is a dead alarm, because high accumulation of p53 causing the initiation of what? Apoptosis signal. Got it? That's another way. So it's instigating the natural mode of tumor suppressor, that is p53, to prevent cancer. Okay. Now drug number 3 or 4, I believe, I don't know. Yes, drug number 3, monoclonal antibody. This is very much important because they ask many questions from this part. Monoclonal antibody mediated treatment. Herceptin is an example of monoclonal antibody which they use for the treatment. Now Herceptin is used for breast cancer treatments, because normally this HER2 is a receptor, epidermal growth factor receptor, or HER2. This HER2 will only activated and function while they form dimer. So dimerization of HER2 is a key event for the cells to grow, right? But this Herceptin is a monoclonal antibody designed to prevent, it's designed to prevent the binding of HER2 to form dimer. So Herceptin is actually preventing the HER2 receptors to form dimer. So as a result, what will happen? No dimerization. So it will prevent the cell signaling, which allows this cells to grow and divide, thus preventing cancer. Drug target number 4. Intermediated signaling molecules are targets. Example, flavopiridol. What flavopiridol does? Sorry. Flavopiridol bound to the CDK2. Binding of ATP to the CDK or cyclin dependent kinase is also very important, because without the ATP, CDK is functionless. Why functionless? Because the CDK is a kinase and they need ATP to transfer the protein to the target, transfer the phosphate to the target protein. So in this case, this flavopiridol interacts with CDK2 exactly in the ATP binding cleft. So as a result, ATP failed to bind and the activation of CDK2 is not possible. And we know CDK are the prime and key proteins for the cell cycle to progress. So that is not possible anymore. Okay. Drug target number 5. The growth factor receptor. Growth factor receptors are always a drug target. We've seen one example, that is Herceptin, HER2. That's blocking HER2 with the monoclonal antibody. And here we are looking at two separate drugs, Glivec and Tarceva. Both these drugs, they inhibit the growth factor receptor, because we know that all this cell cycle, all the growth of the cell ultimately depends on the growth factor signaling that will call the cell, and that will actually tell the cell to divide. For example, the epidermal growth factor, platelet derived growth factor, VEGF, and all this. So these growth factors, if somehow fail to reach the cell, then it will not allow the cells to divide. It will not allow the cell to grow. So in all these cases, Glivec and Tarceva is like a masking agent who can bind with the growth factor receptor and blocks the growth factor receptor. Am I not audible? Tell me, can you people hear me? Yes. So what I was saying is, this Glivec, Tarceva, they are the masking element or masking target. So they can mask the growth factor receptor. So as a result, no matter how much concentration of the growth factor is out there, they are non-functional. Okay. Okay. So these are the different types of drugs that are used to treat cancer, and if any new drug comes in the research, which is also important. So if any new discoveries, any new drugs that are associated to cancer, go through that. It's important to remember. Because I can see that most of you are using this with your smartphone devices, and the sound is not well. Always it's advised to go with a desktop device with speakers for a better sounding experience. Okay, so now let's talk about the third part of this chapter, and that is a host pathogen interaction. Host pathogen interaction, you don't need to read much about the host pathogen interaction, we just read few portions. The interrelations of the host pathogen interaction and some part of it. So host pathogen interaction is just like any other community interaction that we saw in the unit 10. Okay, like, because whenever we are talking about any interaction working in the environment that are either follow these rules of the environmental or community ecology. So you said two species are interacting with themselves. If the chance of getting benefit for both mutualism, chance for getting harm for both competition, benefit for one, harm from another, predation or parasitism. These are the normal situations. So this third one is our choice now, because this chapter we deal with this predation and actually parasitism, not predation. And we know the difference between predation and parasitism. I told you earlier, in parasitism it is causing, it's allowing the host to alive, okay, to stay alive, so that the pathogen or parasites can take advantage of the host. So now, before starting the different part of this host pathogen interaction, you also need to know about few terms: pathogenicity versus virulence. What are the difference and stuff like that. Pathogenicity is the ability to cause disease, while virulence is the degree of pathogenicity. Got it? And keeping this term in your mind, there are two types of pathogens: true pathogen and opportunistic pathogen. True pathogens are all those organisms who will cause disease in any situation. In any situation means who will cause disease in situations like whenever your tissue is infected by them, they will cause the disease. So there is no way that organism is going to stay and take some time to cause, but it will always cause disease. There is no dormancy of it. And there are opportunistic pathogens. Opportunistic pathogens are those who normally are not causing any disease while staying in our body, but only will cause disease in situations where they find the environment to be infection worthy. Okay, that's the difference between true pathogen and opportunistic pathogen. And also there is a difference between infection and disease, although we use it simultaneously, but there is a huge big difference between infection and a disease. Infection is the colonization and or invasion and multiplication of pathogenic microorganism in a host with or without the manifestation of disease, means with or without manifestation of any kind of symptoms. That is known as the infection. So that means the organism is present in our body, it's there, it's colonizing there, it's breaking our tissues, it's multiplying there, but it's not showing any form of symptoms. There is no clinical manifestation. The second situation, or disease, is an abnormal condition of body's function or structure that is considered to be harmful to the affected individual. Okay, and that definitely provides clinical manifestation. Now we can easily differentiate between the two with one example, that is TB. So there are TB infection and TB disease. TB infection, that means a person with a latent TB infection where the TB Mycobacterium tuberculosis, the bacterium is present in our tissue. It's there, but it's not showing any manifestation. How? The person with a latent TB infection does not feel sick, has no symptoms, cannot spread the TB bacterium to the others, and usually has a positive TB skin test and a positive TB blood test. So that is an indication, but there is no further symptoms. That person may have a normal chest X-ray and a negative sputum smear test. So the only way to detect is a TB skin test or a blood test. But a person with TB disease will usually feel sick, weaken, okay, and the symptoms include bad cough, last for 3 weeks and longer, chest pain, weakness and fatigue, weight loss, no appetite, chills, fever, all this. And that person is a reservoir of the disease, so that person can spread the TB disease to the other, and usually has a positive TB skin test and a positive TB blood test, may have an abnormal chest X-ray or a positive sputum smear test. and that person definitely needs treatment for TB disease, although a person with a TB infection does not require any sorts of treatment because there is no disease. So as long as we are clearing out different terms, we must clear out few more terms, like epidemiology is a part of the subject or the field of the study where we discuss about, where we know the origin and spread of a disease across the different places of the country. And few examples if you look at here, endemic, epidemic and pandemic. There are different form of this, a different degree of the disease that is associated. So different degrees, like endemic is, so let me explain that in a small way. Sporadic is the smallest version of it. Pandemic is the largest version of it, depending upon how big that spread of the disease is. Sporadic is the smallest, pandemic is the largest. Sporadic is occasional causes occurring at the irregular intervals. So it's like very few occasions, and intervals are also irregular. That's sporadic, as you can see in this picture. Sporadic means only few occasions and very irregular. So one in this continent, another one is a different continent. That's sporadic. Second type, a little bigger, endemic. Endemic disease, continuous occurrence, but occurrence at expected frequency. Because while a disease start to form, we can first predict the cause of the disease and also try to find out the expectancy of the disease to find the disease in different locations. So the occurrence is continuous, but the expected frequency over a certain period of the time, they follow that expected frequency. Epidemic or outbreak, a little bigger than endemic, is epidemic. Now what happens in epidemic? Occurrence in a community of community or region, and but this occurrence is out of expectation, so above our expectations. So a very huge number of individuals affected, but that's uncontrolled because we cannot predict it beyond expectation. Then finally pandemic. Pandemic involves, like, it's a bigger version of epidemic. Okay, so it's a kind of epidemic involving multiple countries, continents, and affecting a large chunk of the population. That's a pandemic. So if I tell what is an example of pandemic, HIV AIDS is now a pandemic which is traveled across, which has traveled across the continents and now killing million. Epidemic example was cholera and SARS with the coronavirus, and endemic example was malaria in Africa. Still is, it's an endemic. As you see, endemic only occurring in few places, like malaria in Africa is there, and the frequency of this disease is very high in Africa only, but there is also the outbreak of malaria in different places of the world, but not with that high expectancy like that in the Africa. Pathogenesis of bacterial disease. Pathogenesis of disease means the different steps of how a microorganism infects and then spread the disease in the host. That is from the beginning, that is the infection, then colonization, then destruction of the tissue, transferring across the body, and then ultimately manifesting all these symptoms, all these things together, and then come out of that organism and infecting some other healthy organism. All this together is known as a pathogenesis of the bacterial. Okay, so for a bacterial disease, they maintain a reservoir. Okay, the reservoir is always maintained. Now they will move from there to the host and causing the disease, and again come back to the reservoir. So they continue to maintain the reservoir as it is. So maintaining a reservoir, then be transported to the host, adhere to, colonize or invade the host, multiply or complete life cycle on or in the host, and initially evade the host defenses, damage the host, and leave host and return to the reservoir or enter new host. That's a process. Part 1. Acquiring infective agent. Okay, what are the different ways that we can be in touch with that agent?